Welcome students. My name is Dr. Ashok Kumar Mohanty. I am working as a principal scientist at Animal Biotechnology Center, National Dairy Research Institute, Karnal. In the lab itself, when we are doing biology, we are following different aspects of it. One of the most potential area of late has been mass spectrometry. By using mass spectrometry, we can determine the exact mass of a molecule. When we are talking of a molecule, this may be proteins, this may be small molecules in the natural substances, and subsequently we can identify those small molecules with the help of mass spectrometry. As the name as such indicates, mass spectrometry, that means we are going to determine the mass of a particular molecule. Suppose for example, we are talking of proteins. Protein is a polymer of amino acids. As such, the mass of this protein can be determined in two ways. First of all, to determine the intact mass. So, what is the exact mass of this protein? That can be determined by using mass spectrometry. Second thing is, because as such, if you identify the exact mass, it is not possible to know what particular protein it is. So, we have to digest this protein into small peptides, and those peptides will be subsequently introduced in mass spectrometer, and it can be identified by using various softwares. The second aspect is that the other small molecules which are of small molecular size can as such be extracted from the biological samples, which can be used for its identification and quantitation. Module on mass spectrometry is covered under two different parts. This is part A of the module. In part A, I have made efforts to impart preliminary knowledge on mass spectrometry and cover subject matter which is essential to understand part B of the module. So, in the course of my talk, I will speak two different aspects. First of all, basic principle of mass spectrometry, then how we can use this mass spectrometry for identification and quantitation of proteins. So, when we are talking of identification and quantitation of proteins, why we are going to identify these proteins, what for? That means we have a biological mixture of samples, there may be hundreds of proteins and we do not know what they are. So, these days in clinical proteomics aspects, we are talking many a term, like first of all identifying the protein, second thing is discovery of biomarkers. So, in clinical proteomics, identifying various biomarkers for various physiological and biological processes has been a recent trend, where individual biomolecules can be identified and subsequently that can be used for diagnosis of various diseases, diagnosis of various biological disorders. As such, we can identify a complex of proteins which can be further analyzed by using various bioinformatics software. So, first of all, let us discuss the principle of mass spectrometry. What is mass spectrometry? So, study of various molecules based on its mass is called as mass spectrometry, in which a spectra of different masses can be obtained, which can be matched with known masses or pattern of masses obtained from the theoretical breakdown of the whole compound. So, when you talk of the compound, compounds are everywhere. So, the compound is so universal that a wide variety of compounds can be identified and characterized by using mass spectrometry. The first research on mass analysis goes back in the early decades of 20th century, when J.J. Thomson was awarded Nobel Prize in the year 1906 for his creation of prototype to measure the M/Z, that is the mass to charge ratio. So, the concept that M/Z can be studied was first time applied as a tool in scientific experiments to measure the mass of elements by Francis Aston, a physicist from Cambridge in the year 1919, for which he was awarded with Nobel Prize in the year 1922. So, in mass spectrometry, the mass to charge ratio of ions is measured to identify and quantify molecules which are present in various samples. So, in this case, a charged molecule can be deflected under an electromagnetic field and can be forced to travel or spin, which is proportional to the mass of the molecule. Thus, a heavier mass which is charged will travel slowly than a charged molecule which is lighter. The separated ion mass can convert into multiple electronic signals which can be detected by suitable detector. So, I told that different masses will travel through the mass spectrometer. So, these different masses thus can be plotted according to the time which is required to reach to the detector, that is time of flight or TOF, and relative frequency of that mass can be measured in terms of the height of the peak, that is number of unique signals received at a particular time point. From this slide, everything will be very clear as to how these unique signals are received at a particular time point. Next part is instrumentation. Let us come to the instrumentation part of the mass spectrometer. So, basically mass spectrometers consist of three parts. First of all, an ion source, second is a mass analyzer, third is a detector system. So, because our major focus today is to talk on the proteomics and how you are using the mass spectrometry for proteomics applications, we will discuss about the various parts of the mass spectrometer which can be used. Only certain types of mass spectrometers are used in proteomics, for example MALDI, CELD or Electrospray ion sources. Second one, time of flight, quadrupole or Fourier transform mass analyzers. So, what happens when we go for analysis? This mass spectrometer has different parts. First of all is the ionizer, where the sample is kept and this sample gets ionized. After ionization, the sample enters into the mass analyzer, and this in the mass analyzer itself the mass of individual compounds are analyzed, and this traverses through the mass analyzer and hits the detector. After hitting the detector, it gives the signal in the form of a spectrum, and those spectrum are captured by the computer, which are deconvoluted in order to identify these molecules. So, when we are talking of proteins, that means these proteins will be identified. So, therefore, in the mass spectrometry, first of all we will talk about the ionization. There are many methods of ionization. The two main classifications based on energy derived are fast atom bombardment, FAB, which is semi hard. Second one is electron impact, that is a hard method. But for proteomics purposes, first atomic bombardment FAB is most suitable. This includes MALDI, that means it is matrix assisted laser desorption ionization, ESI electrospray ionization, APCI atmospheric pressure chemical ionization, APPI atmospheric pressure photon ionization. Let us come to matrix assisted laser desorption ionization. So, that is otherwise called as MALDI. So, the sample, when we are talking of samples, this is a protein, but the protein has to be digested into peptides, and the sample is mixed with the matrix and allowed to crystallize on a plate, on a MALDI plate. So, the laser is fired at the matrix which produces the ions, and these matrices are 3,5-dimethoxy-4 hydroxycinnamic acid, otherwise known as synapinic acid, alpha cyano-4 hydroxycinnamic acid, 2,5-hydroxybenzoic acid. So, when the laser gets activated, the energy released from here and hits the MALDI plate. So, energy from the laser is absorbed by the matrix, and the matrix then transfer the energy to the analyte. So, in this case the analyte is the peptide. As a result, analyte is ionized into gas phase, and the gas phase analyte travels in the electric field. So, by this way the sample has been ionized. The second ionized protocol is electrospray ionization. Sample remains in the volatile substances, volatile solvent, and the solvent may be acetonitrile or it may be methanol, etc. And these volatile substances are introduced to highly charged needle along with the peptides. This forces the charged droplets from the needle, and solvent evaporation leaves only the charged sample. The purpose of saying is that means the sample in the form of peptides which have been generated or which have been purified are mixed with the volatile substances which are allowed to enter into the source, and the source, when it enters to the source, they are forced to be charged droplets which comes out of the tip of the needle, and subsequently they evaporate leaving the charged samples. So, now we have prepared the ionized substances, but once these substances or samples have been ionized, they have to enter into the mass analyzer where the mass of the samples will be analyzed. This is the segment of the instruments which separates the ions based on its m/z value, that is mass by charge ratio. These are of many types. Time of flight is one, quadrupole analyzer, it has low resolution but it is fast and cheap. Ion trap mass analyzer, it's good, it has good resolution, all in one mass analyzer. Another is magnetic sector analyzer which has high resolution, determines the exact mass. Ion cyclotron resonance, these are the highest resolution, it produces the exact mass but it is costly. These different type of analyzers can be put in tandem also continuously in various combinations and modification, thus getting different nomenclatures like triple quadrupole, MALDI Q-TOF, Q-Q-TOF, ESI Q-TOF, LC ESI, MS, MS, etc. Now, let us discuss about the mass analyzer time of flight. So, in case of time of flight, ions are mobilized by high voltage and this travels through the flight tube, and while traveling they are deflected by the reflectron and ion mirror which increases the path length, often it may lead to doubling and therefore increases the resolution, and the time taken to reach the detector is directly proportional to mass of the analyte. So, that means when the ions they travel through this MALDI tube, the TOF tube, it hits the ion mirror which subsequently increases the resolution, and because of which the resolution increases and the time taken to reach the detector is proportional to the mass of the analyte as I told before. So, now let us discuss about various mass analyzers little more in detail. First one is the time of flight. So, the time of flight, that is otherwise called as TOF, it is capable of differentiating molecule based on its molecular weight. The ions liberated from the ionizer and accelerated through the vacuum in mass analyzer are separated based on the m/z ratio. It is because different size molecules take different time to reach to the detector. The whole system operates under intense vacuum during the entire procedure. The signals which hit the detector are amplified, the ions are subsequently analyzed. So, the relative abundance of the ions is interpreted on the basis of their mass by charge ratio and respective intensity. So, it is high throughput technology and there is no upper m by z limit for the detection. So, TOF has been modified according to the type of required application, that is TOF in triple quadrupole is good for amino acid sequencing. MALDI QTOF is good for amino acid sequencing as well as molecular weight determination. QTOF is good for amino acid sequencing and any protein modifications. So, a quadrupole mass filter consists of 4 parallel metal rods with different charges. 2 opposite rods have an applied positive potential and the other 2 rods have a negative potential. The applied voltages affect the trajectory of ions travelling down the flight path. At specific voltage, specific entities reach to the detector while others collide to the rods and fail to reach. Therefore, it is possible to select different m by z entities by scanning the sample over a wide voltage range. It is different from the time of flight in the sense that here the ions are separated mass analyzer by voltage, while in TOF the ions are separated by difference in the time of flight. So, in triple quadrupole instrument, 3 quadrupoles are added in tandem. The middle one is for the fragmentation, usually based on the CID. The first and the second act as the mass analyzers. With triple quad, we can analyze various samples. We will talk about this later. The third most commonly used ion trap mass analyzer. So, ion traps are ion trapping devices that make use of a 3-dimensional quadrupole field to trap and mass analyze ions. It was invented by Wolfgang Paul, who won the Nobel Prize in the year 1989. It offers a good mass resolving power. It is used to trap ions in a confined space so that MS and MS-MS analysis both can be used in the same space. Different ion trap devices have been developed like 3D ion trap, linear ion trap, orbit trap, etc. So, when there are many mass analyzers, can we put different type of mass analyzer in one mass spec and try to analyze our samples? The answer is yes. So, that is otherwise called as Tandem MS. What is a Tandem MS? Different mass analyzers are kept in tandem. It is done basically to fragment the precursor ions. What is a precursor then? That means when a particular substance which has been ionized enter into the mass spec, it has different chambers. In the first chambers, in the mass analyzer itself, the mass is analyzed and it gives a mass. So, the mass which comes out in the first instance can be called as a precursor, and subsequently these precursors ions are broken down in subsequent mass analyzers which gives daughter ions. So, in a mass spec principle, either you can get the intact mass, and subsequently if intact mass, that molecule has to be again broken down to give some daughter ions which will give smaller mass, but they have been derived from the precursor's mass. So, different mass analyzers are kept in tandem in a tandem mass spectrometer. It is done basically to fragment the precursor ions, and they may be combined in different combinations like quadrupole, magnet sector quadrupole, quadrupole time of flight, time of flight. So, in this all ions are separated by fragmentation. What is fragmentation then? That means when the molecule is entering into the mass spec, it has to be broken down into smaller elements, smaller peptides. So, multiple methods of fragmenting take place by various methods. One is collision induced dissociation, that is CID, electron capture dissociation ECD, electron transfer dissociation ETD, chemically assisted fragmentation CAF, and this is a part of the mass spectrometer. So, students, till now we have discussed about the principle of mass spectrometry. So, just to give an overview of this. First of all, we have taken the samples, then those samples have been prepared, which have been generated into small molecules. These sub-molecules are introduced into the source of the mass spectrometer, and the source ionizes the molecules, and subsequently this enters into the mass analyzer. In the mass analyzer itself, the masses of those spectra are determined, and subsequently it is captured by the detector, and the detector deconvolutes. From the detector, it is captured in the form of the mass spectra. So, when you have so many mass spectra, you will be simply confused what to do with it. So, what we do? Then we have to use various softwares. There are inbuilt softwares these days which deconvolute these mass spectrometric data into different mass. Suppose one particular molecule or few mixture of various molecules have entered into mass spec. They get differentially charged. They may be monoisotropic charged, they may be multiply charged. If they are multiply charged, in that case what happens? There may be different spectrum, but they are having same mass because they have been charged differentially. So, we have to analyze the data. When you talk of analysis of the data, what does it mean? That means first of all, we have to deconvolute all the mass spectra into monoisotropic spectra, and then subsequently those spectra has to be used for identification of the molecules. So, coming to the data analysis, mass spectrometer generates data in the form of list of ions, that is called as compounds, which are detected in the order of various mass to charge ratio, that is M/Z. For proteomics purpose, this M/Z needs to be transformed into all possible peptides. Data analysis is therefore of two types mainly. First of all, identification of the compounds. Second, differential quantitation of the compounds in two or more samples. So, when the spectra are generated, they show the fragmentation of the peptides, which are used to determine the sequence of those peptides by a search algorithm. It may be any other compounds, they may be metabolites as well, and subsequently its structure has to be determined. You are required to refer part B of the module on mass spectrometry for acquiring complete knowledge.